Hi, my name is Isabella Jimenez, and this is non-Mendelian and Mendelian genetics under 2 minutes. Let's go.
Here's some key vocabulary terms to note before we begin. An Austrian monk named Gregor Mendel in the 1800s carried out studies on pea plants to study the inheritance of traits. He first crossed different varieties of purebred pea plants, and then grew the resulting seeds to determine their characteristics. He then crossed the offspring with each other and then grew the seedling. His studies showed that characteristics could be masked in one generation but reappear in later generations, and proposed that inheritance involved discrete units of inheritance from one generation to the next. Based on his studies, Mendel's conclusions were that organisms have discrete factors that determine its features. Organisms have two versions of each other, gametes only have one version of each factor, and one version of an allele that is dominant over another, and will be completely expressed if present. Certain rules can be established from his conclusions, which include the law of segregation. When gametes form, alleles are separated so that each gamete only carries one allele for each gene. The law of independent assortment. Segregation of alleles for one gene occurs independently to that of any other gene. It is important to note that the law of independent assortment does not hold true for genes located on the same chromosome. Linked genes. More on that later. Lastly, the law of dominance. Recessive alleles will be masked by dominant alleles. And it is important to note that not all genes show a complete dominant hierarchy, so some genes show co-dominance or incomplete dominance. Moving on to these exceptions in non-Mendelian genetics. Not all crosses follow Mendel's laws. As I mentioned earlier, linked genes do not follow these laws. They are genes carried on the same chromosome and as a result do not sort independently. A common example of this is the gene for wing size and for body color in fruit flies. They are located on the same chromosome, and certain body colors and wing sizes usually occur together. In Mendel's trials, the traits he studied in pea plants were associated genes that were on different chromosomes. Other non-Mendelian inheritance patterns include codominance and incomplete dominance. In codominance, both alleles are fully expressed, whereas in incomplete dominance, neither allele masks the other, producing a blend of the two parental phenotypes. Moving on, multiple alleles are genes with more than two alleles in the population. For example, the ABO blood system have the alleles A, B, and O. People can be any combination of the two alleles. Lethal alleles can be lethal in the homozygote, heterozygote, and may be dominant or recessive. One example of this is the Huntington's disease. It's an autosomal dominant disease, meaning that you only need one copy of the gene to get this disease that damages nerve cells in the brain, causing them to stop working properly, which can affect movement, cognition, and mental health. Next, sex-linked genes are genes located on the Y-X chromosome. Color blindness is a good example of this because it is a sex-linked recessive trait located on the X chromosome. So color blindness is more prevalent in males than females because males only have one copy of the X chromosome, therefore not having a second copy to possibly mask that trait. Multiple genes are when one phenotypic trait is determined by more than one gene. Skin color, eye color, height, and hair color are all polygenetic traits. Hypertrophy is when one gene influences two or more unrelated phenotypic traits. One example of this is sickle cell anemia. This genetic mutation results in sickle-shaped blood cells and also leads to other effective traits. And lastly, non-nuclear inheritance. Some DNA can be inherited via mitochondria and in plants via chloroplast. This DNA is inherited from your mother, and similarly, in the chloroplast, it is inherited from the female parent in plants. To sum it up, Mendelian genetics can help us derive genetic patterns, while non-Mendelian genetics are occupied with the patterns that do not follow these rules. Thank you for watching.